I'm happy to see so many of you have joined us. Today I'll be giving you the webinar on complete and unbiased genetic characterization for the QC of transgenic animal models. I am Nicole Hendricks. I'm the International Sales Manager at Genentech for anything genetically engineered models. Today we have the webinar section, and afterwards we have some time for a Q&A. You have a screen on the right-hand side where you can add any questions during my talk or afterwards if you think of anything. Of course, I'll also give you my email so you can ask me any questions afterwards. So today I would like to show you what kind of added value our TLA technology would have to your research. I'd like to begin by showing you some examples of what we have been able to do for others in the past. I think you're all familiar with, or perhaps even work with, transgenic mice, which is one of the most powerful and versatile tools in mouse genetics. It's commonly used due to the control it gives on timing and location, so this might seem like a foolproof way to integrate genes and structural changes. However, a great range of concatenamers can occur. And as we were able to show for Genentech in the paper shown here on the left side, several unexpected changes have occurred. And not only can our TLA technology provide you with the insight that there has been structural changes, we can even provide you with details to a single nucleotide level. For Jack's laboratories, we were able to observe co-integration of E. coli sequences in 10 out of 40 animals. So not only were we able to help Genentech and Jack's, we currently have a great number of published papers, including 25 in the field of transgenic animal models, as well as several other applications like cell line development and cell and gene therapy. So to be a bit more exact in what TLA can offer in comparison to other conventional techniques, I'd like to draw your attention to this table. I assume you're all familiar with these well-known techniques and what they have to offer in terms of identifying genetic variants. For example, FISH is commonly used to determine integration sites and whole genome sequencing to check for any concatenamers. However, all the techniques shown above come with their disadvantages. So if the intention is to sequence the entire genome, then whole genome sequencing is the way to go. But when you know exactly which transgene you're using, or already know which genes you are specifically interested in, then generating such large volumes of data is rather an inefficient way to go about it. It wouldn't be time nor cost effective and would only complicate the interpretation of your data. So yes, a combination of these techniques can provide you with good results, but this is often time-consuming and expensive process. So using our TLA technology, we can not only provide you with information on a single nucleotide level of structural variants, we can also provide you with a clear and complete overview of all of these genetic variants. So as you can see here in this table, we are able to reliably output all of these different characteristics that are of interest to our customers. And which is also good to know is we do this for a competitive price starting at 1,450 euros, the short turnaround time, which can be as low as 4 weeks. So nucleotides that are close to each other in the 1D genome are of course also close in the 3D genome. TLA is based on this knowledge of this proximity. So by following the steps you can see on this slide, TLA enriches all nucleotides that are close to each other in the 3D space. When we receive a sample, we start by cross-linking them, and for this we use formaldehyde as a cross-linking agent. After we proceed to fragment it using restriction enzymes. After this, we fragment it with religation. This forms circular TLA templates, as you can see in the middle of the screen. This is a stochastic process yielding millions of cells. The next step is then the enrichment step using transgene-specific inverse primers that face outwards. TLA can construct complete integration sites, as you can see in the bell-shaped curve at the bottom. this happens in a complete hypothesis-free fashion. So in the end, we will generate a very high sequencing coverage across the transgene. The coverage will be highest in the vicinity of the primers that have been used, and the coverage will decrease further away from these primer pairs. And as you can see here, due to our hypothesis-free way of working, we can provide you with the exact locations of the integration, as well as the number of integration sites. And just to briefly talk you through the pictures, on the vertical line, you can see the chromosomes, and on the bottom, you can see the chromosomal position. So what you see here on the left is that we were able to determine a single integration site on mouse samples. And on the right plot, we found multiple integration sites in a goat sample. The peaks you see in this genome-wide coverage plot are the bell-shaped curves I showed you in the last slide. As I mentioned, our inverse primers bind specifically to the transgene, and therefore we will be able to provide you with the location of the integration site in an unbiased manner. And of course, we do not only look at these genome-wide coverage plots. Using next-generation sequencing, TLA will obtain fusion reads of the integration sites, and with this be able to provide you with information on a single nucleotide level. We will not only be able to provide you with coverage across the transgene, but across the flanking regions as well. So these fusion reads are, of course, an ideal solution for exact PCR-based genotyping assays. So therefore, I also briefly wanted to draw your attention to this slide. Usually when designing genotyping primers, these will be placed on the breakpoint sequence between the genome and the transgene, like you can see in the pink arrows. And to test for heterozygosity, the primer pairs in green would be based on a wild-type genome sequence. Of course, this works in the instance of a clean integration, but what if a duplication has occurred? Here, the primers would bind to your duplication as well as your transgene, giving you a false positive result for heterozygosity. Why I am showing you this is to show you how our TLA-based solutions are able to attack these exact structural variants to design an exact genotyping strategy. So a great example of what our TLA technology can do, you can find in this new paper recently published by the NIH. The transgenic line they wanted to analyze has become an important tool in adipose tissue biology over the last few years. However, the exact genomic transgene integration was not established. Using TLA, it was possible to locate the transgene on chromosome 9 between exon 6 and 7. And not only were we able to detect the location, we also managed to find a transgene-transgene fusion. So as I was able to demonstrate in my previous slide, TLA not only reliably detects integration sites, we also provide you with insights on any concatenamers that may have occurred. As you can see here, we can show any transgene-transgene fusion on a single nucleotide level and provide you with the orientation of the fusion as well. As you're probably well aware, several different transgene-transgene fusions can occur, including partial deletions or integrations. This can lead to a faulty expression of the transgene and is therefore of great importance to analyze and be aware of. TLA can reliably determine structural variants in the transgene and genome. Here you can see the standard bell-shaped curve that I've showed you in the slides previously. In case of structural variants, you will see a very characteristic plots as these. And of course, in each of these different situations, we will look at the fusion reads as well to provide you with more in-depth information. So as I mentioned before, TLA looks at the transgene on a single nucleotide level. Therefore, we are also able to reliably provide you with the detection of single nucleotide variants. On the left here, you can see the occurrence of frequent single nucleotide variants, which could possibly indicate the mutation has already occurred in the development of the transgene. And on the right here, you can see a great example of infrequent single nucleotide variants, which points to different things, but for an example, to the presence of different copies of the transgene being integrated. I think we can all agree this is key information in your research. Below here, you can see a table of how we output these results. I will talk you through it to make it a bit more clear of what kind of information we provide you with. On the first row, you can see on position 301, A is mutated to a G. We were able to determine this in 100% of the reads. Which is also interesting to note here is that you can see under the sequence coverage that each position is sequenced over hundreds of times. I talked to you about this recently published paper by the NIH a few slides previously, and I wanted to briefly come back to it. What I didn't mention before is that this paper, they talk about a back sequence. And you might be wondering if TLA might still work on a sequence this large. And actually, the short answer is yes. With TLA, we can look at any type of integration on a single nucleotide level. So back integrations as well. For example, here you can see a human back integrated in a mouse genome. We identified the integration sites on chromosome 9. And of course, you could have done this using fish as well. But what is interesting to see here is the peak you see on chromosome 8. This shows there is a 57 kb co-integration from chromosome 8 on chromosome 9. This would not have been detected without using TLA. So just to briefly summarize what we have talked about, I've told you about how TLA can detect structural variants and co-integrations. We can provide you with whole genome coverage plots. We can also look at all of this information more in depth by looking at any concatenamers, at the breakpoint sequences, and any sequence variants. So transitioning from transgenics to genome editing, I now just wanted to briefly take you back to another paper. I think everyone here is aware that genome editing in mice using either classical approaches or using CRISPR-Cas have been reported to harbor off-target effects. For example, off-target integrations or gene segment duplications. I've now showed you that we can provide in-depth insights in off-target integrations. But what about knockouts? In this paper, we were able to show a tandem duplication where deletion was expected. Whilst I've shown you that TLA can be used to completely characterize transgene integration sites, it is also well suited to elucidate the outcome of target gene editing on specific locus. Hereby, rather than designing the TLA primer pairs on a transgene sequence, the primer pairs are designed to specifically amplify the region surrounding the edited locus. As we generate two independent data sets, any and all structural variants in this locus will be detected, again in an unbiased manner. A great example of this is shown here, where the locus on chromosome 12 was edited with CRISPR-Cas9. Two primer pairs are placed adjacent to the editing sites, thereby sequencing out from both sides. Looking at these genome-wide coverage profiles, you see two translocations occurred, which could be resolved completely with TLA. Resolving such structural variants with, say, a PCR approach would be impossible unless there was already prior information on the potential fusion partners. So we've talked quite a lot about insertions and concatenamers, but what about deletions? Our TLA technology is also able to exactly pinpoint the deletion by using our two independent primer pairs. But what most of you probably are aware of is that the deleted region can also reintegrate somewhere else in the genome. What we can then do is design a third primer pair. This primer pair we design is specific to the deleted region to pinpoint exactly where this fragment has integrated. So I briefly want to end up with a little bit about us at Genentech. We were founded in 2012 and we're based in Utrecht, a historical city in the center of the Netherlands. We have around 35 employees of 12 different nationalities. We provide our DNA technology on services as well as kits. With this, we support our customers in various applications such as the genetically engineered models, cell line development, and cell and gene therapy. Thank you so much for your attention today, and I hope I gave you a clear overview of what our TLA technology has to offer and how we work. Please feel free to submit any questions on the panel on the right, and I'll be back in a few minutes to answer these. If you would like to have a more in-depth discussion or about your questions or about what TLA does, please feel free to send me an email on the address on the slide. I will now just make an inventory of the questions and I'll be back to you shortly.